so wonderful to be in the Bahamas and to be working with all of you, because I think together we can build a great path forward. And a lot of people wonder how you would use gene therapy, a really powerful technology to change lives. And I'm going to go over that and actually how we can make it affordable right here in the Bahamas, which is a very important part of the equation and is not talked about very much. My story begins with being a patient, not myself, but my son. From 2011 to 2013, I volunteered my time for the advocacy of the use of stem cells. I actually got to talk to a lot of people in this room, although they might not remember me. We were trying to learn what they were doing, how they were doing it, and how to actually translate that to the world so that people would support stem cell use and we could get embryonic stem cell refunded. But in the meantime, I didn't know that there was a disease brewing in my child. And in 2013, we ended up in the hospital. Where were all these treatments I had learned about? Nothing was really available. We're still treating type 1 diabetes with insulin, the 6th most expensive liquid in the United States, even though it costs pennies to make. This is old technology, and the technology had come much farther than this, and I knew that. And not only that, the other patients in the hospital needed an advocate. So I went looking for cures for kids, and I became a patient. I ran into aging research, and I found out that basically a lot of genes that can actually help us extend our healthy lifespan can also treat childhood diseases. And to this day, my company, BioViva, every gene we choose will treat also a childhood disease. So we can help both ends of the spectrum. We can help these kids get cures and then not just live another decade longer before they start dying of the diseases of aging. So we're going to step back a little bit because most audiences need to understand why we would use such powerful technology to treat biological aging. Non-communicable diseases are now the biggest killer on the planet. They kill 74% of the population. And the top diseases are all aging-associated diseases. So by actually looking at aging as a disease itself, it gives us the ability to make the biggest impact in the world. That means, actually, when you put the numbers in, about 41 million people will die of these diseases, and we often don't take these very seriously. I know everyone in this room has seen this, but for purposes of people who might be watching online, we like to go over what these big killers are and what they actually look like statistically by age. And this is why we believe that biological aging is, in fact, a disease. Because if you were to take any one of these diseases here and cure it, you'll die of the other ones. They're all running in parallel. They're a cellular process that is running within us. And we have the ability to actually tackle it upstream, to do therapies that we might only need to do once or twice in our lifetime. We don't have to go back in twice a year to take therapies. We may be able to actually put a human, eventually, into homeostasis, meaning that they regenerate just a little faster than they degenerate. And why would we do this? Everyone in this room might also know that. It's the silver tsunami is coming. Economically, we cannot afford the aging population to continue to decline. There are not enough payers coming into the system, and we need to keep these people healthy and agile. And not only that, it has to do with the terror response to death. Come on, let's face it, these are not just numbers. I don't want to get dementia. I don't want to not know who you are. You know, I don't want to die of cancer in a ward. I don't want to die of cellular senescence, like happened to a lot of people who died from COVID, short telomeres in their immune system. These things are treatable, and we need to start working on this now. So again, lifespan and health span, what are we trying to do? We're trying to extend your health span, but we're going to try to extend it as far as we possibly can. Try to put human bodies into homeostasis so that they can live viable, wonderful, active lives. And this is a graph that shows the curve of human aging and death. And this is really important to take into consideration. I think it was Richard Swift that once pointed this out, that this curve isn't just your friends leaving you and your parents dying and your grandparents passing. It's actually loss of returns. So, you know, as Richard would say, you make a lot of money, and right when you finally go to enjoy all of the rewards that you have reaped, you actually have a physical downturn in your ability to actually do anything with that money that's meaningful, that you can really feel and enjoy. And this is a new graph that I'm showing here. This is from the cost of aging. And this one really actually startled me. Not much startles me anymore after 10 years of studying death. But this is something that I really want you to take a close look at. So this is labor income, and this is the consumption. So actually your early life, which makes sense, into your 20s, you consume more than you make. That makes sense. Your parents are paying for you. But at this point, you actually drop below the line. How many of you still have dreams after 60? How many of you would still like to buy a new house after 50? This would tell you that most of the population cannot do that. That just like 1950, you should be preparing for death. You better stay in the home you had your children in. You better not have dreams, because you are going to consume more than you make very soon. I reject that. So, what is gene therapy? Man, Bill did a great job of talking about all this stuff that's happening in gene therapy. What does it look like? How will you experience it? When you experience gene therapy as a patient, it's just merely an injection. What we do is we take what were once viruses, we basically neuter them so they can no longer divide, they cannot deliver their genetic material. We trick them into delivering healthy strands of genes. Human genes that are associated with the upregulation of beneficial proteins, making your cells more youthful and younger. This is the genes being delivered directly to the nucleus. When you hear about gene therapy, you hear, basically it's almost under an umbrella. There is somatic cell delivery, which is what we excel at. It's been around for decades. We take genes and we deliver them into your cells, and they have long-term transcription of proteins that change you. CRISPR is also gene therapy, and it's delivered the same way with neutered viral vectors. And when you hear about editing, you're hearing about some sort of in-between of these technologies. But those are all gene therapies, and they're very exciting. I always like to show a video of gene therapy in use. This was a study that was done by Pfizer. The boy on the left has muscular dystrophy, and that means that his muscles are atrophied. He's not really able to do much physical activity. That's him trying to climb stairs. This is him on the right 3 months after gene therapy. He may only need to take this gene therapy once in his life, but more likely maybe 2 to 5 times. That's a huge impact for sitting down and taking an injection. So my company, BioViva, we work in AAV and CMV, but there's somebody in your audience that also works with some of the plasmid-based therapies, making more affordable therapies, and that's something that we're not going to focus on, but if you get a chance to talk to Mac, you should talk to him. So how do we know that gene therapy works? You know, this sounds all very hypothetical, but in fact it's not. These are all of the approved US FDA gene therapies. They have their trial path through what's called monogenic disease, single gene mutations, and these are all now drugs that you can get. And so we know that the technology works. It can take patients who have life-threatening diseases and take them off of long-term treatments for the rest of their life. The first patient for severe combined immune deficiency, or boy-in-the-bubble disease, who was treated some 40-some years ago, still talks publicly about the benefits of gene therapy on their life. So this is not new technology. It's actually been around for a while, and there's a lot of fascinating research around it. When we look at treating biological aging, though, we're going to have to go to the hallmarks of aging. So you need something to measure whenever you're going to treat something that is probably polygenic, meaning it's not a monogenic disorder. It's not like hemophilia A. In this case, it's the whole system disrepair. So when we look at the hallmarks of aging, that essentially gives us some biomarkers in which to go by. So when we pick gene therapies, we're going to pick them specifically for trying to reverse processes of these hallmarks. If you're not familiar with these are the processes that are happening at the cellular level. This was the first agreed upon group of them. The group is growing now. There is an expansion of the hallmarks of aging, and it will continue to do so. It's kind of like our laundry list of things that we want to change that are changing with aging. In some cases, you may not need to change all of these hallmarks. By changing one, you can actually affect another. When we look at human telomerase reverse transcriptase, this is a gene therapy that has extended the lifespan of model organisms many times. Our lab at Rutgers University extended the lifespan of mice by 40%. If you use the scale that Bill Falloon was using with the OSK factors, that's 140%. Every researcher does it different. But this gene itself, this single gene that basically came with a big worry package from the oncology group, has never been shown in longevity studies to accelerate or make cancer active, okay? This is a gene that cancer uses, like hundreds of other genes in your body, to actually immortalize itself. But when it's delivered to cells, it actually protects them against cancer. When we have short telomeres, that's when we're most likely to get cancer. That's called genomic instability. And genomic instability is one of the targets of age-TERT. Another one is stem cell exhaustion. If we had like a super one wish we could make in gene therapy, we'd say, with this gene therapy, could we just target all the stem cells in the body? And that would be great, because it would sort of superpower them for more cellular divisions and more healthy replications in which we had a better epigenetic reset. Of course, it directly affects telomere attrition. This is the one and only route that I know to definitively lengthen your telomeres without actually killing bone marrow cells and just getting new cells into your T lymphocyte stream. And we showed that it significantly restored mitochondrial dysfunction. I'm going to list our papers after the talk, or lower in the talk, so that you can look them up and you can look up all the data. If we wanted to just go for mitochondrial biogenesis, we would choose a gene called PGC1 alpha. It actually helps restore mitochondria. It turns what they call white fat into brown fat, and actually researchers tell me that's because of the visual amount of mitochondria that are actually re-established in the cells. And this might be a great treatment for obesity. When we look at gene-based senolytics, there's a lot of companies that are working on different variations of removing senescent cells. So we hear that a lot in our industry, wanting to remove them. Gene therapy is a very, very powerful way to do that. And there's a little bit of a cautionary tale around that, because it will remove senescent cells very quickly from your system. It's a very, very effective treatment. Partial reprogramming. Bill went over that very well. There's very many variations of that. And as you know, we want you to work with reputable companies and all, but this is all open sourced information. This is the most magnificent time in history in which you can actually go read these papers yourself and you can find out what are in these therapies. Of course, we hope that you come to our companies to actually develop them for you. Now, there are probably 25 other genes that we work with, but I just wanted to put a few on here that actually support healthy aging and mechanisms that we may not directly associate right now with the hallmarks of aging that they probably are. Follistatin, which we had already talked about, you saw the boy climbing up the stairs. Follistatin is probably the lowest hanging fruit for everyone in this room. We hope that it will protect you against sarcopenia, help you after you've taken cancer therapies with cachexia. It protects, in literature review, against several types of cancer and protects your liver. This is a powerhouse. It blocks myostatin, allowing your body to continue to gain muscle mass as you age instead of becoming more frail. FGF21 is a superpower house for cardiovascular disease and is being used after patients that have had heart attacks. Clotho is a superpower gene therapy, and it is associated with better cognitive performance, protecting your kidneys against kidney failure and cardiovascular disease. And then SIRT6 is an epigenetic regulator, basically keeping your cells epigenetically in a stasis rather than letting that epigenetic drift change you, making you older, making you more frail, and devastating your body. I have taken 4 of these gene therapies. I'm happy to say we've shown safety since the company was first developed in 2015. I have helped hundreds of patients get access in extreme situations to these type of technologies. I've found doctors who will help give these technologies. I've helped over 20 people get into clinical trials in the United States. A lot of people don't even know how to find a clinical trial when they have a devastating disease. I am 100% committed to creating this genetic revolution. So when we look at gene therapies that we could do right now for biological aging, this is sort of the list. This is the lowest hanging list of what we can do now. So we can deal with telomere lengthening with gene therapy. We can increase your muscle mass. We can enhance cognition. In our studies with Clotho, we saw a standard deviation improvement in IQ scores. For high-powered people, that's a difference between, phasing out and staying into business for another 10 to 15 years. For low cognitive people, that could make the difference of being able to function in a normal way in society. So we're really excited about that. Kidney protection is done by the same gene. Mitochondrial therapy, we have, again, several technologies that work with that. Follistatin helps with mitochondria as well. Telomerase reverse transcriptase and PGC1 alpha. Nerve regeneration, this is a new one that we're working on now for the optic nerve, for glaucoma. And then, of course, epigenetic reprogramming. So these are all low-hanging fruits to move gene therapy into treating biological aging. So how would we make this affordable? This is really why I'm here. This is a big build-up to just this. So in order to make these technologies affordable, we need to work in a few areas. First of all, we need to see what's already been done and what went wrong. I don't know if anyone has followed. Has anyone in here followed the price of gene therapies? Okay, we've got 2 people. Well, that's probably good because I'm about to really scare you. These are all the approved gene therapies, and this is their price in the United States. So if your child is born with spinal muscular atrophy, there's actually kind of a lottery on it, whether it's going to be $2 to $5 million, or else they generally die within 6 weeks of birth. This is unsustainable, and I want to tell you what went wrong. When we started with monogenic disease, we created an unaffordable therapy. If you start with a disease that has a very small pool of people, investors, VCs, everyone wants to get their money back. And what you create is a very expensive drug. Gene therapy manufacturing itself is still very expensive. So you're looking at a difference between 3 million people to 56.3 million people in the US alone who are over the age of, what is this one, 65. I've got varying statistics. So right here, we see that this is a market that could bring these prices down significantly. You have a big market. This is what we want to see in drug development. We do not want to see small markets. And when you manufacture in larger quantities, each dose is less. So when we looked at medical tourism, we found that they could get the cost of each dose down by 10% if there were if there were about 20 patients, and up to 50% if there are 1,000 people or more. So now we're starting to get down into price range that could make gene therapy more affordable to more people. When we look at the cost between what we consider medical tourism or an emerging zone where we could charge for gene therapy and an approved gene therapy, look at the differences. So the cheapest gene therapy in the United States is $425,000 to treat one eye. We could do that outside of the US for $60,000, as safe, with all the efficacy. And actually, when you work outside of a regulatory body that gives you the right, once you're approved, to do a lot of different things, you will see the level of safety go way up. The level of caution goes way up. The level of risk the doctor holds goes way up. We have worked with medical tourism companies since, I think, about 2016. We've never seen an adverse drug event that was severe. And yet, in the United States, through the FDA, there have been 6 deaths from known doses that were toxic. They were in the literature. They shouldn't have been done. So what are we doing to make gene therapy even more affordable? So my company now is working with a cytomegalovirus. It's a larger virus than AAV. We started in AAV. Bill Falloon mentioned AAV. It's a fantastic little tenacious virus. The problem is that every time you have a gene of interest, you have to buy a whole therapy. So if I want to take the 4 gene therapies that I took, which I did, I have to buy 4 distinctive therapies. Each one of them could cost over $100,000. They could cost much more than that. With CMV, what we're trying to do is package multiple genes into one delivery so that we can deliver on a fraction of the cost. And if you read our paper in PNAS, you'll see that we delivered it both intravenously and intranasally, and it was redosable. And this is something we have a big problem with AAV. We have become experts in redosing AAV, but CMV would be the better bet if the research translates to humans, as it does in animal studies. This year, we're going into a non-human primate study, and so we'll be excited to show you what happened in that in probably about a year, and these will be old macaws. So here's our peer-reviewed studies. So this is the intranasal and injectable gene therapy. That's in PNAS, and that is with CMV. This was for dementia. 5 patients with dementia, 4 out of the 5 got cognitive benefits. 1 of them got long-lasting benefits. This was a massively low dose of gene therapy. It was a dual gene therapy intranasally delivered. The last one is the age-related disorders, and that's my gene therapies. That's my 4 gene therapies and what happened to me over a multitude of years. So why we need new medicine now? Why do we need new places in order to launch this technology? Well, today's prescribed drugs are old. We've been over that. Anything that you can be prescribed today in the United States is at least 15 years old. Innovation is working at a breakneck speed. We cannot afford to take these drugs. And these drugs, these old drugs, come with 100% mortality. We know everybody dies taking them. We need new options. We need to pioneer. We need to pick up a new spirit. We need to pick up a new space race, a new trip to the moon. And we need to get people excited about pioneering new technology. There's a lack of innovative treatments in the regulatory zone. We know that. We've been around. We have human data. We have brought human data to the US FDA, and then they say go back and do a few more $1 million of animal data. Small companies like ours can't do that. We just can't. And we're here to help patients, you know. And so, you know, that doesn't make sense. And the animal data is just honestly non-predictive of outcome. There are a lot of things that we take today, you know, some antibiotics, aspirin, that would never pass a phase 1 study in animals. You know, they're actually toxic to them, and yet they revolutionized human existence, penicillin being one of them. And new and more precise technologies lack funding. We need to get that funding in. We need to turn over new technologies, and like we discussed earlier on the panel, we need to be able to shift midway and say, we think we might even be able to make this better. So who would be the stakeholders of this? Literally everyone. I could have just put everyone. But number 1 is the patients. We always have to think patient first, because as I mentioned in the beginning of this talk, you will all become a patient. So making sure that you pave the way of dignity to your own treatment is essential. The medical doctors need the right to use new medicine. The doctors that we see that go offshore to do medical tourism type technologies are like the most amazing people in the world. They're people that have said, I am not going to keep killing my patients with these drugs. The do no harm does no longer fit here because we have new options and we need to try these new options. Otherwise, they're failing their patients. Governments could save a lot. I think one of Sinclair's papers showed that if you just slowed aging by 1 year, it was literally the savings of what, like $38 trillion for the US? And that doesn't even account for our personal loss, taking care of loved ones, missed work, the extra cost of putting people in nursing homes, and the actual, emotional cost of watching this happen in front of us. Investors could come to zones like these and they can find out how drugs work before having to put millions of dollars into them. They can help people get safe access, ethical access, and find out if a drug will work for a fraction of the price. The failure rate through the US FDA is 94%, and that's just phase 3. Most fall out long before that, usually and typically because of lack of funding. So I want to thank you so much, and I hope you enjoyed sort of a fast go through gene therapy.